going to talk about Schwann cells. When we talk about Schwann cells, we should know that those are principal glia of the PNS, peripheral nervous system. They are the supporting cells of the PNS. Now we are going to present a video about Schwann cells development.
Axon. Schwann cells start to develop in the embryo and continue to increase the wrapping around the axon through childhood. This development increases the thickness of the wrappings, which peaks in adolescence. This is why teenagers have such quick responses. The Schwann cell contains the typical cell organelles and cell membrane structure. However, notice as the Schwann cell surrounds the axon that the nucleus and other organelles are squeezed to the outside wrapping of the cell. This outer wrapping of the Schwann cell is called the neurolemma. The inner lining is made up of layers upon layers of cell membrane. This inner wrapping is called the myelin sheath.
Now we have Schwann cells regeneration. Schwann cells are known for their roles in supporting nerve regeneration. Nerves in the PNS consist of many axons mediated by Schwann cells. If damage occurs to a nerve, the Schwann cells will aid in digestion of its axons, phagocytosis. Following this process, the Schwann cells can guide regeneration by forming a type of tunnel that leads toward the target neurons. The stem of the damaged axon is able to sprout, and those sprouts that grow to the Schwann cell tunnel do so at the rate of approximately 1 mm per day in good conditions. The rate of regeneration decreases with time. It's really important for us to know about types of Schwann cells. First, we have myelinating, which are wrapped around axons of motor and sensory neurons to form myelin sheath. And also we have non-myelinating, which are cells involved in maintenance of axons and are crucial for neuronal survival. Another subject that we're going to treat in this presentation is the immunoreactivity. The different classes of Schwann cells spread characteristics and genic markers that can be targeted with antibodies. Myelinating Schwann cells can be visualized by immunohistochemistry using antibodies against the proteins X100, myelin protein 0, and myelin basic protein. In the other case, non-myelinating Schwann cells, such as those that form remake bundles and terminal Schwann cells, are positive for X100 and glial fibrillary acidic protein. The next video is going to talk about action potential.
To the axon. One advantage is the regeneration of severed axons. Another advantage is an increase in the speed of the propagation of the action potential along the axon. The rest of this presentation will concentrate on the increased speed of action potentials down the length of the myelinated axon. Here is the neuron, and you can see the repeated Schwann cell membrane forming the myelin. Note that there is a small space between the Schwann cells where the axon is not covered by the neuroglial cell. These spaces are called nodes of Ranvier. From what you already know, action potentials occur at the axon hillock and continue to be repeated away from the cell body, much like dominoes falling one after another.
Now that we know that we have two types of Schwann cells, myelinating and non-myelinating, we have to consider thick myelinating advantages, which are basic to regeneration of several axons and increase in speed propagation of action potential along the axon. The next video that we are about to watch is going to talk about myelinating axon versus non-myelinating axon.
Here the unmyelinated axon with the myelinated axon. You can see that action potential reached the end of the myelinated axon more rapidly than the unmyelinated axon. The speed of the propagation is faster going from node to node than action potentials that develop adjacent to the previous action potential.
Applications in medicine. A number of experimental studies since 2001 have implanted Schwann cells in an attempt to include remyelination in multiple patients. In the past 2 decades, many studies have discovered positive results in the use of Schwann cells as a therapy for spinal injury. According to applications in medicine, now we have a Schwann cell clinical trial. The first step is generating all of the preclinical safety and efficacy data that justify the testing of Schwann cell transplantation in humans has been completed. That first step was a major undertaking, however. The first step in the clinical trials process is to conduct a phase 1 trial, which is strictly focused on safety outcomes. That is particularly important in our situation because of two factors. First, we are talking about putting cells inside the spinal cord, and second, we are sticking a needle directly into the spinal cord in order to deliver the cells. Finally, we have a video that is going to summarize what Schwann cells are. And that's all for this video. Thanks for your attention.
Schwann cells surround the neurons of the PNS. The axons of peripheral neurons are often surrounded by cells of another type, known as Schwann cells. These cells are important for providing support and nutrition. They also act as an electrically insulating sheath. The individual cells are interrupted by gaps called the nodes of Ranvier. Because of the insulating effect of the Schwann cells, signals travel along the axon in a series of jumps. This process is also called saltatory conduction. The signal jumps from node to node.